So today our topic will be apoptosis. So first of all, what is apoptosis? So it is a programmed cell death. The body kills the own cells whenever there is any disturbance within the cells, or if the body feels like the cells are no longer useful or even harmful to the body. The term is derived from a Greek word which means falling off or dropping off. It is a coordinated and genetically controlled process. It is very important for the normal development and tissue homeostasis. What do you mean by tissue? Yeah, having a similar function. Having the same function, the group cells are called tissues. So these are some examples of physiological apoptosis. Why is it physiological and not pathological? No, only pathogens mean pathological. No, not like that. Like physiological means it is done by the body itself and there is no other agent or anything else involved in this. Therefore, it is physiological. So these are examples. One is death of neurons during CNS development. What do you mean by the CNS? Means what does it comprise? What things constitute the CNS? Yeah, brain and spinal cord. So during its development, there is obviously growth of the cells. There is formation of new cells in the CNS, and there will be old cells. Now, it is not that when the baby is born, that baby will carry the cells up to 80 years old. Not like that. Nah? Obviously, the baby will lose the cells, or the person during the course of development will lose some cells. So those cells are lost by apoptosis. So new cells get generated, and the old ones get degenerated. Then there is degeneration of many tissues like web between fingers during fetal development. So in the fetus, if you look for it, you can find out that there are web-like features in the hands or in the appendages in the hands and feet. Some web-like features, but these are not usually available when the baby is delivered. There are no webs between the fingers or the toes. So these are also removed by the body. So how it is removed? It is by apoptosis only. Then there is apoptosis of blood cells like eosinophils. Eosinophils are also important for the body, but sometimes when they are not required, the body does apoptosis and kills off these cells. So this process helps remove unwanted, harmful, or even self-reactive cells. Sometimes the cells act against the own body or own organ. So in those cases also, if the body is functional enough, the body will act and kill the cells by apoptosis. Then what is the significance of apoptosis? One is it maintains tissue homeostasis, means the tissues are maintaining a balance. It prevents accumulation of abnormal cells in the body. Obviously, if abnormal cells get accumulated in the body, it will be harmful for the body in one hand. And another thing will be the body will get less efficient because there will be storage of debris, storage of unwanted things in the body. So body will obviously not keep this waste. So it degrades these things with the help of apoptosis. Then decreased apoptosis can cause malignancy and autoimmune diseases. What do you mean by malignancy? Not only tumors. If any tumor or something like that, if it spreads to the body, if it is growing at a very rapid rate, then it is called malignancy. Then decreased apoptosis can cause all these things. Autoimmune diseases can be caused. Then malignancy may be caused. And if there is increased apoptosis than the normal rate, there will be neurodegenerative disorders. So I already gave you an example that during the CNS development, there is the destruction of the neurons due to apoptosis. So if this destruction increases beyond the control of the body, obviously there will be degeneration of the neurons and the CNS will be affected. So these are the neurodegenerative disorders. Then there is another term called necrosis. You will come through it, I think in pathology or in physiology only, you will come through this term. So first of all, what is the difference between apoptosis and necrosis? So apoptosis is programmed and it is energy dependent. It is programmed, I already told you in the class, that this is done by the body itself, so it is programmed. And energy dependent because the body is giving effort to do all this. So obviously it will record energy. So it is energy dependent. There is no inflammation because there is no external agent working in the body or any autoimmune condition like that. So there is no inflammation. Then there will be cell shrinkage and nuclear fragmentation. I have an image by which I will show you all these things. The cell will shrink in size. Shrink means, it will get slim, or it will just opposite of swelling up. It will shrink, and the nucleus inside it, or the DNA, will get fragmented, will get broken into pieces. So these are the features of apoptosis. Then what are the features of necrosis? It will be accidental or due to any injury, like burn or anything like that, which causes inflammation in the body, which is an external thing in the body, which is not it is actually accidental and something which is not intended by the body, maybe due to injury. Then necrosis is it causes inflammation. Whenever there will be necrosis in part of the body, any cells of the body, any tissue or any limb, organ, so there will be inflammation accompanied with the necrosis. In this case, it is just opposite. There will be cell swelling and membrane rupture. The cell will swell, and due to pressure, the membrane will rupture, and thus the cells will get destroyed. So this is a diagram, a simple diagram to differentiate between how necrosis occurs and how apoptosis occurs. So you can see that in necrosis, in this part, the cell is little bit swelled up, but here it is shrinked. And after that, you can see that there is the organelles are taken inside blebs, like a little bit of debris. They are containing the organelles. The organelles are not just split apart; they are contained within some bodies. But here in necrosis, you can see the organelles are getting differentiated. There is nothing like any structure is not covering them. So this is the basic difference. Then after that, morphological and biological features of the apoptosis. So firstly, there will be cell shrinkage. Shrinkage, I have shown you in the picture. The cell will decrease in its size. Then there will be chromatin condensation. The chromatin, that is the nuclear material, that will get condensed. Then there will be formation of apoptotic bodies. These are the bodies, like this. These structures, they will contain the important parts of the cell. So these are called apoptotic bodies. So this will be formed. These things. There will be no inflammation because it is already done by the body in a systematic manner, in a coordinated manner. So there will be no inflammation. There will be proteolysis, means breakdown of proteins. DNA fragmentation, there was in the picture, the DNA was getting fragmented, it was getting broken into pieces. And exposure of phosphatidylserine. There is a compound that is also exposed when the cell is broken. Here is another picture which is showing apoptosis. Have a look at this picture. So these are the normal organelles. This is the cytoplasm of the cell. This is the cell membrane. Then here you can see the cell membrane has changed the shape, and the cell has got significantly smaller, which is shrinkage. After that, the membrane is blebbing. Blebbing means the different organelles are getting packed into some other structures like vacuoles. So these are called apoptotic bodies. After that, these bodies are individually phagocytosed by phagocytes. Like phagocytes are large cells which eat up these bodies. So these are not wasted, or the body can use this to make other cells also, or the body can break them down to make energy, so that nothing is wasted. Up to this much is clear. Then there are two mechanisms of apoptosis. I told you that already apoptosis occurs by the body only, but there are different reasons why apoptosis will occur. It is not like that suddenly the body will waste its energy. Body will never waste its energy to destroy anything it has created. Again, it has to make. So there are some pathways due to which this apoptosis occurs. So one is triggered by activation of caspase. Caspase means there are some proteins which trigger the breakdown of the cells, or the cell membranes, or the altered cells. So these are in many steps they will be activated. Firstly, these caspases will be in inactive form, and due to some reasons, by following some pathways, these caspases will be activated. The two major pathways are intrinsic, mitochondrial. Mitochondria, you know, na? Mitochondria, it is in the structure of the cells. So it is an internal part of the cell. So intrinsic means nothing else from outside the cell is involved, or the cell itself is responsible for this apoptosis. And one is extrinsic, there is depth receptors. Something else comes and binds to the cell membrane or wherever it can bind, and there is a programmed cell death, means some agent external from the cell, it is coming and just helping the cell to perform this apoptosis or triggering the cell. Now there is some complex steps, just you can see it is in your book on Injectable, it is already present. So first there will be internal or external stimuli, because for anything stimulus is required. The cell will not suddenly break down its own body without any reason. There is stimuli. Then there is FAS receptor activation in cytotoxic T cell. There are some cells, cytotoxic T cells means these are necessary for the body for defense mechanisms. Sometimes these cells will recognize any other cells of the body which are harmful to the cell, to the body, or which are no longer needed. Then these T cells will have some receptors. Those are known as FasL receptors. I have not mentioned it here. So they will come and bind to the surface of the cells. There is something called ligand. So I'll tell you later. Then there is ceramide generation and activation of caspase. I told you there is a protein called caspase which is responsible for breakdown of the cell. Then there will be mitochondrial injury, then which is followed by DNA damage. Then formation of nuclear protein TPP3, which induces cell death protein, which is called BAX. So there is some complex mechanism involved here. Just try to get the overview what occurs in this. Then there will be cytoplasmic and chromatin condensation. I have already shown you in the picture of this apoptotic bodies. Then there will be apoptosis. The different organelles will get form blebs and they will keep on separating. Then there will be phagocytosis. As I have shown you in this picture now, there will be phagocytosis going on this cell. So this is some detailed picture of one is intrinsic, another is extrinsic. Like look here, intrinsic, nothing else is involved. But in extrinsic, you can see that there is FAS TNS receptor. So receptor ligand interaction. This is ligand, this is receptor. This comes and binds, and then this will trigger the breakdown of the cell or the apoptosis. And here there is nothing triggering it. The mitochondria itself is doing it. Just for your reference, I have been given this picture. Then we have gone into little bit detail of the intrinsic pathway, how the mitochondria helps in the apoptosis. So it is not normally triggered. Normally the mitochondria will not just go and destroy the cell. What happens is when there is trigger by the cell itself due to like DNA damage, or sometimes DNA will get damaged due to radiation, toxins. That's why people are told that we shouldn't go near very much radiation or any toxic substances like nuclear radiation. So these things will affect the DNA, or not necessarily nuclear. Any type of radiation may damage the DNA. So in those cases, what happens if the body finds out that the cell's DNA is damaged, or if there is oxygen, a lack of oxygen called hypoxia, or there is cellular stress due to like free radicals or many things are there which can cause cellular stress. So if there is any damage to the DNA, then the mitochondria will release certain proteins in the cytoplasm, which are mainly Cytochrome C and SMAC. These are different types of proteins. These are very complicated if you go into details. So then Caspase 9 is activated. Caspase, I told you, these are proteins which take the role of the apoptosis. Then these are actually in inactive form. But whenever there is any stress or there is any indication to cause apoptosis, then these are activated by different mechanisms. So in between there are many more steps. I've just given few of the steps to make it more clear. Then Caspase 9 itself will activate the Caspase 3. This is another cell. So another enzyme or protein. So these on activation will cause apoptosis. Now there should be two mechanisms in the body. If it happens that only apoptosis is going on, the person will not live. Every cell will die. So there must be a mechanism by which it has to prevent also. So there must be a balance. So there is a protein called BCL2 family proteins, which regulate whether mitochondria will release cytochrome C or not. So means simply this BCL2 family proteins will regulate the apoptosis, whether the apoptosis will occur or not. If these are activated, then the apoptosis will not occur. then sometimes this may cause apoptosis, like that. There is some balance. Now extrinsic pathway. I have shown you in the picture now. The extrinsic means something else will come from outside the cell, it will bind to the cell surface, and that will trigger the apoptosis. So it is triggered by ligands. What is a ligand? A ligand is a molecule that binds to specific receptors. There will be a specific receptor in the cell. There are many receptors. One ligand will bind to the specific receptor only. So that's why it is called ligand. This is that molecule that will bind to a specific receptor. So ligands are like FasL or TNF. There are many more ligands. So these are just examples. So there will be some receptor called death receptor, because obviously these receptors are only used when there will be the death of the cell. So these ligands will come and bind with these receptors. These will activate caspase 8. Pro-caspase 8 to active. Pro means it is not active, it is inactive caspase 8. Then it will activate the caspase 8. Caspase 8 activates other caspases like caspase 3. I have already told you that caspase 3 directly causes apoptosis. So this is the this was the intrinsic mechanism. This is the extrinsic mechanism. Then there is regulation of apoptosis. Obviously, I told you this has to be regulated. Otherwise, the body will just shut down due to continued apoptosis. So what are the regulating mechanisms? There must be balance between pro and anti-apoptotic proteins. Pro means the proteins which will cause apoptosis, and anti means obviously it will block apoptosis. So what are the pro proteins? These are BAX, BAK, and anti-apoptosis proteins are BCL2, BCL, XL, MCL1. Mediators are, these are the names: BAD, BID, BIM, PUMA, NOXA, NOXA. Then P53 protein promotes apoptosis after DNA damage. So these are the some of the proteins or some of the things which control the apoptosis, which can either increase or decrease the rate of apoptosis in the body. So again, these are some steps how this occurs, apoptosis occurs. FAS receptor activation, then cinnamate generation, then DNA damage by ROS, cell shrinkage and condensation, formation of apoptotic bodies, and phagocytosis. Now these are apoptosis is actually very important, and these are the physiological causes why apoptosis has to occur in the body. Otherwise, it will become pathological, or it will become troublesome for the body. So one is endometrial shedding. So you know that during ovulation there is increase in vascularity or the thickness of the endometrium, because it has to contain the egg. It has to provide nourishment to the egg. But after some time, when the egg is not fertilized, the endometrium has to shed. Otherwise, the body will not keep on feeding it now without any reason. So obviously, there will be endometrial shedding. So shedding will not occur just casually. So there is apoptosis involved in this. One is regression of lactating breast. So lactating breast, during lactation, the breast size will increase to feed the baby. So breast size will increase. There will be changes in the breast. But after some time, like after 2 years or more, when the baby is not taking breast milk, so obviously the breast size will decrease slowly with age. So there is regression of the lactating breast. This is also done by apoptosis. Like then there is thymic involution. Can anyone tell what is thymic involution? Any idea? Thymus? Thymus, you know, na? What is thymus? What is that thing? Specifically, gland, thymus gland. There is a gland called thymus gland in the neck, which sits in the neck. But in adults, it is not that much prominent. So it is more active when the person is small. Then during
age के साथ है.
Then when the person becomes adult, slowly and gradually the thymus will reduce. बोल रहे हो? Because the body no longer needs it. That's it. Then intestinal epithelial turnover. Then intestinal epithelium also gets changed due to this apoptosis. Then there are some pathological reasons. The body is not causing it. The body is causing the apoptosis, but the body is not solely responsible for this. So what are the causes? One is infection. I told you now that pathology doesn't always mean that only pathogens will be involved. Many other things. Fire also can be involved. Any accidental material can be involved. Radiation can be involved. So one is viral infection. In hepatitis, the liver is affected. The liver will slowly degenerate. So that is induced by the virus. That is one type of pathological apoptosis. One is radiation injury. I already gave you an example. it causes DNA damage. So obviously any cell's DNA it can damage and there will be apoptosis. Then there is degenerative CNS diseases. Like I've given some examples like Alzheimer's, major diseases are there. One is chemotherapy-induced cell death. We use this for our own benefit. Like in cancer patients, we use this chemotherapy. We give some chemicals to the patient's body which will destroy the DNA of the cancer-specific cells. Sometimes it damages the normal cells also, but mainly it is aimed to destroy the cancer cells. So this is chemotherapy-induced cell death. This is also apoptosis. Then morphological and pathological changes, pathophysiological changes. What are the changes which occurs during apoptosis? One is it affects single or few cells. It may affect one cell also, it may affect some group of cells also. Whatever the body needs, feels important, it will do. So there is the cytoplasm will be eosinophilic, then nucleus will be condensed. Membrane bound apoptotic bodies will be formed, but there is no inflammatory response. Because only due to only one cell, there will be no inflammation, no? And it is already done by the body. Then there are some biochemical changes. One is proteolysis of cytoskeletal proteins. Cytoskeletal means cyto means cell, skeleton means the skeleton of the cell, the structural composition of the cells. So some proteins will give structural support to the cells. So these will get lysed, means the cell membrane will get dissolved. That's why it will shrink. So first step is this. There will be proteolysis of the cytoskeletal proteins. Then there will be cross-linking of the proteins. There will be fragmentation of nuclear chromatin by activation of nuclease. Nuclease, I think you have studied in biology, na? Nuclease enzyme, there is one enzyme which damages the DNA or the chromatins. So this enzyme will be activated and then that will cause fragmentation of the nuclear chromatin. A glycoprotein molecule called thrombospondin and a phosphoprotein molecule called phosphatidylserine appear on the outer surface of the apoptotic bodies. The bodies are formed, so they appear on the outside surface, which facilitate recognition by macrophages for phagocytosis. So macrophages will come and eat them. So obviously for recognizing them, this thrombospondin and phosphatidylserine, they are required for identification. Then apoptosis is occurring in the body. So a person who has come to you with increased apoptosis, like a disease is there which is called due to increased apoptosis. So how will you recognize it? So there are some techniques by which we can understand whether the apoptosis is increased in the body or it is normal or not. So one is DNA fragmentation assay by electrophoresis. You know na, that by electrophoresis we can know whether the DNA, we can know the shape of the DNA, then how much is getting fragmented or not. So by electrophoresis we can know it. Then one is called tunnel staining. One is by demonstration of chromatic condensation by hematoxylin and eosin. So these are some stains by which we can demonstrate the chromatic condensation. Then there are other stains called fuelgen, acridine, orange staining. Then estimation of cytochrome C. So I have already told you in the class that cytochrome C is released by the cells, by the inside of the cell to destroy the remaining part. Which then activated caspase. Obviously caspase is normally in inactive form. So we will find out the activated caspases, the number or the amount of activated caspases, and annexin 5. All of these are required in this apoptosis. Then disorders with altered apoptosis. I already told you that there should be a balance, that it should be neither too high or not too low. So what happens if it is very low? Then malignancy. Cells will do whatever they like. So cells will get increased by their own because there is no safety mechanism for the body. Then there will be autoimmune diseases. The various cells will come and attack the other organs of the body because the body is not that efficient to destroy those cells. On what happens in the reverse case, like if there is very much increased apoptosis, one will be an Alzheimer's disease, Parkinson's disease, Huntington's disease. What is common in these diseases? Can anyone tell me? Cell death is common in everything, na? Already we are discussing about cell death. Huh? These are neurodegenerative diseases. These are CNS diseases. So the person who is affected will have some or other CNS effect. Like the person will have forgetfulness or many things, many symptoms are there. So these are actually CNS degenerative diseases because there will be increased CNS destruction. Then there might be myocardial infarction because there will be damage to the heart or to the blood vessels. then there will be stroke also. Obviously, same reason, due to the damage of the blood vessels, there will be stroke. Then this is the summary. So apoptosis is a programmed energy-dependent cell death due to proteins. Caspase will increase it, VCL will control it. Then occurs by intrinsic and extrinsic pathways. Dysregulation leads to diseases, as I told you, that the increase and decrease both can cause diseases. Then detection aids in diagnosis and therapy. I told you different types of detection methods like different gene electrophoresis, like that. Those are used to diagnose the disease, the condition, and for therapy, whatever is required. So it ends the discussion. Thank you.